Hi guys, my name is Ashley Smith. Today is July 27, 2021. It is 6:20. Today we will, this is actually Lab 6, Exercise 1, biochemical testing for microbial identification. I have my setup here. I disinfect the area, applying my PPE on, and now I'm ready to get started with our exercise. So here I have the E. coli bacteria, and then I have my E. coli labeled broth along with the epidermis and then the labeled broth as well. I have my two pipettes and sterilizing in alcohol, and of course my lit candle. So we're going to start with the E. coli first. I'm going to uncap it, and I'm going to place that, and I'm going just to sterilize the lip of the E. coli. And I'm going to place this here. I'm going to uncap my broth, and I'm going to just sterilize the top. Okay. And I'm gonna take my pipette out of my alcohol, being sure that all the alcohol is out. I am going to grab my E. coli vial, inserting the pipette without it touching the edges. And I'm just going to grab some sample here. And I'm gonna place 4 drops into my broth. 1, 2, 3, 4. Okay, place that there. I am going to sterilize my lip again prior to putting the lid on. And I'm going to sterilize the lip of the E. coli. Okay, that is it for this sample. We will repeat the same steps for the epidermis sample, and then we will place both samples in our incubation location for 48 to 72 hours, wait for growth. And then we are going to, at that time, we're going to do the methyl-red and Vogus Prosker testing. And we are going to take pictures of our slides and upload it to our data table. So that is it for exercise 1. Like I said, we're going to repeat the same steps for the epidermis. In the meantime, we're going to move to exercise 2. This is lab 6, exercise 2. This is the biochemical testing for microbial identification, the catalyst testing here. So once again, I sterilize my area. Of course, I have my PPE on. Here I have my inoculation sticks, sterilizing in alcohol, my pipette and my hydrogen peroxide. I have my cultured agar plates of E. coli and epidermis. I also have my labeled slides of the E. coli and epidermis. So at this time, we're going to flip the E. coli over, and then we're going to take the inoculation stick, being sure that there is no alcohol on there. So we're gonna take our cap off of our plates, we are going to get a sample of our agar from our agar plates. And we are going to place the sample right into the center of our slide here. Okay, place that to the side, and now we are going to apply 1 drop of our hydrogen peroxide onto our slide. And if we have any bubbling, that is an indication that any oxygen bubbles, that's an indication that it is positive. If we don't get any bubbling or if we don't see any oxygen bubbles visible on the slide, that is an indication that it is a negative sample. So now we're just gonna apply our 1 drop there. And I do see some bubbling. Okay, and there I have some oxygen bubbles there. So it looks like we got a positive culture test, and we are going to again repeat the same exact steps for our epidermis slides, and that will complete this exercise too. Thank you.